Welcome to part 1 of 3 on chapter 16. In this chapter, we're going to be looking at simple patterns of inheritance or genetics. So inheritance is defined as the acquisition of traits by their transmission from parent to offspring. So basically how our genetic material is passed on and how those traits are passed down from one generation to the next. So from the beginning of human civilization, we've noticed that our offspring look very similar to the parents. And we also see this for lots of other organisms as well. So in the 1960s, again, we didn't know about DNA yet, so they had this hypothesis that there's kind of a blending inheritance happening. So your traits from your two parents were blended together to create that next generation. But in the 1950s, when we were learning about chromosomes and DNA and the genetic material, that's when we had this particulate inheritance, where there were these particles that contain the traits that were being passed down from our parent to our offspring. And now we know these particles were in fact chromosomes that had DNA that contained all these genes that coded for all sorts of different proteins, and then those proteins gave us our traits and characteristics. So in this chapter, we're going to look at it. There's 5 different topics. The first part of this chapter, we're looking at Mendel's 2 laws of inheritance. So Mendel, and I'll talk about him more, he came up with these 2 laws of inheritance that is basically the very start or the very simple part of inheritance or genetics. And then we'll get into the chromosome theory of inheritance. So this is an actual theory that supports this idea of inheritance. And then we'll look at a few little neat twist on Mendel's 2 laws. So we'll see a little, some fun little things to go through in the third part of this chapter. So Gregor Mendel, he was a monk in Austria in the 1800s, and he was enrolled in the University of Vienna, and he took a lot of physics courses, a lot of math courses, and he also took a lot of natural history courses, which were biology courses during this time. So after he took all this math and science, he became a monk, and so he had a lot of time on his hands as a monk. So in the 1850s, specifically 1856, he started to do genetic experiments in his garden because he really liked working with plants and studying all the different traits of plants. In 1966, so 10 years later, he actually published a paper from all these experiments that he had done in the previous years, and he titled it Experiments on Plant Hybrids. So Mendel, his paper was ignored at the time, which is very unfortunate for him because he never was around to actually receive credit for all of his really hard work and his ideas, and that later became laws. And he was actually quoted as saying, "My scientific work has brought me a great deal of satisfaction, and I am convinced it will be appreciated before long by the whole world." So he knew that his work was very, very important, but the world just wasn't ready for his ideas quite yet. And this unfortunately happens to a lot of scientists because people get stuck into certain ideas, and it's really hard to change your thoughts about different things. So in the 1900s, after there was more research done on genetics and people started to look at chromosomes and to kind of really get into this topic, that's when his paper was rediscovered. And now it is basically the foundation for all genetics that we do today. So he is a very, very important person to help start inheritance in genetics. So what he did was he worked in his garden. He worked with plants, specifically he worked with garden peas, which you've probably seen growing in a garden if you parents have gardened or your grandparents have gardened or you've been around gardens at all. And the important part about garden peas, there's several properties to them that make them really good plants to do these experiments with. So one property is that there's lots of available characteristics or traits. We're used to peas being green. The pea pods are green because that's how we see them in the store. That's how they look more appetizing to us. But in reality, garden peas have lots of variation in different traits. So here are 7 different traits that show variations. So one is flower color. The flowers are pretty big, so it's pretty easy to tell what color the flowers are. They're either purple or white. The flower position, either the flowers are along the stem, so axil, or they're at the top of the plant, and those are called terminal flowers. Seed color, we know peas can be green, they can also be yellow. Seed shape, we usually see these nice round seeds, but they can also have a wrinkled shape to them. The pod shape can be smooth or it can be constricted. The pod color can be green, which is the one we're used to. They can also be yellow. And then plant height, you can either have tall plants or there are shorter dwarf plants. So you can see there's lots of different characteristics that Gregor Mendel could look at and follow as he crossed different plants together and collected data and went from one generation to the next. Another advantageous property is that normally garden peas are self-fertilizing. So plants are neat in that a lot of plants have both male and female parts on the same flower. So this means that the flower can fertilize itself. So the female gamete or the egg can be fertilized by the male gamete or the pollen with the sperm in it from the same plant. So it's really easy to get what are called true breeding lines. And true breeding lines just means that the parent looks exactly like the offspring. It has the exact same characteristics. Another good example of true breeding are domestic dogs. If we breed a Dalmatian to another Dalmatian, we expect all of the puppies to look like Dalmatians. If all the puppies look like Dalmatians, that's considered to be a true breeding line. And it's the same with any other domestic dog like golden retrievers. You breed a golden retriever to a golden retriever, you expect all of the puppies to be golden retrievers. So again, that's a true breeding line. All of the offspring look like the parents. And here's an example of the flower, and you won't learn about flowers until a biology class later on if you continue into the biological sciences. But if you look at the flower, so this is a cross section up here. The flowers, they have ovaries, which are in the middle of the flower, and then in the ovary they have ovules, which are eggs for that. And then up at the top of the ovary you have something called a stigma. This is where the pollen attaches, and then the pollen produces a pollen tube. The sperm travels down the tube and fertilizes all the ovules. The pollen in the sperm is created on the stamen, which is the male part of the flower. So if you look at a flower on the stamen, you have these little yellow structures, and then that's where the pollen is located. So really big flowers, and you have both male and female parts on one flower. Because these plants are normally self-fertilizing, it's really easy to make controlled crosses between two different plants. And again, this is because we have really large flowers, so it's easy to see what we're working with without a microscope. And then you can actually pick two plants and you can do a cross-fertilization or create a hybrid plant between your two known plants. So this is really nice because you can actually track who the parents are and what types of offspring that those parents produce. So in order to do this, it's very, very easy. In your plant, you take one plant and you're going to remove the stamen from our purple flower up here. So you just take a scissors, cut off all the stamens so that that flower can no longer produce any pollen. And then what you do is you have a second plant, so we have a white flower on the right hand side. We're just going to take a paintbrush, pick up pollen from that white flower, and then transfer it to the stigma of the purple flower. Then that pollen will then fertilize that purple flower. So now we know the female was the purple flower, the male was the white flower, and we know that we cross them together using that paintbrush. So very, very simple to create these hybrid plants. So what Mendel did was, remember we had all those 7 different characteristics that Mendel is looking at. He just took one of those characteristics, and we're going to take plant height. So plant height, you can either have tall plants or you can have dwarf plants. So what he did was he performed what's called a monohybrid cross, or a single factor cross. This is where he just followed one variant of one characteristic or one trait. So again, we're just looking at plant height. So what he did was he took plants in a P generation. These are true reading plants. So he took a plant that was tall. If that tall plant self-fertilizes itself, all of its offspring will be tall. And then he crossed this with a dwarf plant, which is also true reading. That dwarf plant, if you allow it to self-fertilize, it's going to produce all dwarf plants. But in this case, he took the tall plant and the dwarf plant, and he did that cross-fertilization that we just talked about. So that's called the P generation, or the parental generation. The offspring that were produced are called the F1 generation because they're the first generation from your P generation. So your offspring are the P cross. And again, we're looking at monohybrid, so we're just looking at one trait and how it's passed down. So in this example, when we cross a tall plant with a dwarf plant, all of the offspring end up being tall. Okay, so this is pretty interesting because what happened to that dwarf characteristic? Did it just disappear? Is it still there? That's the thing that Mendel was really interested in. So what he did was he took that F1 generation and he allowed them to self-fertilize themselves. And then he counted up the characteristics of the F2 generation. So your F1 generation self-fertilizes itself. You collect all those seeds, you plant them, and then see what those offspring grow up to look like. And what he found was that out of all of the offspring in the F2 generation, 3 of them were tall, or 75% of the plants ended up being tall. The other 25, or the 1 plant, ended up being dwarf. Okay, so you can see that that dwarf characteristic, it wasn't lost in the F1 generation, it was just hiding, and it shows up in the next generation, the F2 generation. Okay, so what Mendel did was he took each of our 7 characteristics and he did this exact same experiment. He collected lots and lots of data and then figured out this ratio that we see at the bottom, the 3 to 1, 3 tall to 1 dwarf. So here's his actual data. So he was a very good scientist. He took really good notes. He had very large sample sizes. If you look at the very top cross, we looked at flower color, so purple and white flowers. Going across, he counted about 1,000 flowers, and 700 ended up being purple, 225 ended up being white. So he got about a 3 to 1 ratio for flower color as well. If you look at the very bottom trait, this is the tall and dwarf stem, so plant height. These are his actual numbers, so 787 ended up being tall, 277 ended up being dwarf. So he got this approximate 3 to 1 ratio. So for each of these 7 traits that he looked at, that he experimented on, he found out that all of them showed this very, very close 3 to 1 ratio. So there's something going on. So like any good scientist, he didn't just, say, hey, I got this 3 to 1 ratio. He actually went beyond that and he tried to explain how you could get this 3 to 1 ratio. So from this experiment, 3 important ideas came out of it. So one was that there is a dominant trait and a recessive trait. So each of our characteristics we looked at, remember they had 2 options. So flower color either had purple or white flowers. Plant height, you could be tall or dwarf. So one of those is dominant and the other is called the recessive. So the dominant is the trait that is displayed more often. The recessive trait is displayed less often because it's being masked by that dominant trait. And this can explain why one of those traits shows up a lot more over the other trait. So why we see purple flowers more over white flowers, or why we see tall plants more than short plants. So this is his first really important idea from this experiment. The second important idea was that it has to do with genes and alleles. So remember, at this time we didn't know about genes or DNA, so he called them particles. So he came up with this particulate mechanism of inheritance. So he said that there's some kind of unit factors that are being passed down from parent to offspring, and now we know that these particles or these unit factors are in fact genes on our chromosomes. So he stated that in order to get this 3-to-1 ratio, every individual has to have 2 genes for every characteristic. And these 2 genes, if they have variant forms of each other, they're called alleles. Okay. So here we're looking at plant height. For plant height, you can have 2 different forms, or 2 alleles. You can either have a gene for a tall plant or a gene for a dwarf plant. So because both of those genes code for plant height, they're considered to be the same gene, but they're just variations of each other, so we call those alleles. And the third idea, this is where his law comes into play. He says that these 2 alleles in every individual, they are segregated or separated from each other. So the 2 copies of the gene carried on our F1 plant are going to segregate or separate from each other so that each sperm or egg carries only 1 allele. And this is called Mendel's law of segregation. It's his first law. So your 2 copies of your gene are going to segregate or separate from each other during the transmission from parent to offspring. And if you think about the previous chapter and meiosis, when we create the eggs and the sperm, this makes sense because remember, we all have pairs of chromosomes. We have homologous pairs of chromosomes. Those homologous chromosomes are separated from each other during meiosis 1, and then that creates gametes that only have 1 copy of every chromosome. So here we're kind of pulling the 2 chapters in this unit together because they relate to each other. So meiosis is how this actually occurs. This genetic part is looking at how those traits are actually passed on and how we get our different characteristics. Okay, so we kind of talked a little bit about alleles. In order to do your problems in your homework and on the exam, we have to look at a few more terms. So we talked about alleles, which are alternative forms of a gene. We can also look at something called a genotype and phenotype. So the genotype is the genetic composition of their individual. So genotype genes. So think about the word and what it means. So the genotype relates to what genes you have or what alleles are in that individual. Okay, so if a genotype of an individual, and here we're going to represent our different genes with letters to make it a little bit easier. So if our tall allele is the dominant allele, we're going to represent it with a capital letter. So our tall allele we're going to represent with a big T. So if your individual has a big T, remember you have to have 2 copies of every gene, so we have a big T and a big T. We have 2 tall alleles. That individual is called homozygous dominant. So homo means the same, dominant means the dominant trait, so we're going to have 2 dominant alleles for the genotype. The dwarf trait we're going to represent with a little t. So if you have a genotype of little t, these individuals are called homozygous because the 2 letters are the same, and they're recessive because they're both little. So you can also have homozygous recessive genotype. The other genotype you can have is a big T, little t, and these are called heterozygous. So hetero means different, so our two alleles are different from each other. We have a big T and a little t for this. And then based on this genotype, we can actually determine what our individual will physically look like. And that's called the phenotype. So phenotype, physical characteristics, so the characteristics that are a result of gene expression. The big T, and big T, little t, so those two genotypes, the homozygous dominant and the heterozygous, because they have one allele that's dominant, those individuals are going to show the dominant characteristics, so they're going to grow up to be tall. If you have a homozygous recessive, a little t, because both of the alleles are recessive, we're going to end up with a plant that has a dwarf phenotype up here. So genotype and phenotype, make sure you really know these terms, and then the homozygous dominant, homozygous recessive, and heterozygous, because you'll have to use these in our inheritance or genetics problems that are in the homework and your exam. In order to solve these genetic crosses that we're going to be doing, we're going to be using something called a Punnett square. So Punnett square is developed to help us predict the outcome of different genetic crosses. So what we're going to do is we're going to take our F1 plants up here. So remember, all of the F1 plants ended up being tall. But if we really look at it, their genotypes, they have to be big T, little t, and we're going to cross it with another plant that has a big T, little t. So here's how you solve these genetic problems. So step 1, you're going to write down the genotypes of the parents because you have to know what you're starting off with. And I gave these to you. In the problems, you may have to figure them out. The problem may say the male is heterozygous for plant height, the female is heterozygous for plant height. Remember, heterozygous means that the two alleles are different from each other. So we're going to have a male that is big T, little t, and a female that is also big T, little t. So now we know what we're starting off with. The next step is to figure out the gametes that each parent is going to make. And this is where we use Mendel's law of segregation and our ideas of meiosis and how the gametes are created. So our male gamete, we're going to just separate the big T and the little t. So one sperm cell has a big T, the other sperm cell has a little t. And just to kind of pull the previous chapter together, we started off with a cell that was diploid. It had 2 T's in it. Now we have cells that are haploid. They have 1 T in them. The female gamete, because her genotype is the same, she's also going to produce a gamete with a big T and a gamete with a little t. Okay, so now we know what kinds of eggs and sperm cells we have. So then what you do is you put these gametes onto your Punnett square. And just for convention, across the top of the square we put the male gametes, so we have a big T and a little t. Down the side we'll put our female gametes, we put a big T and a little t. So you create this empty square, you put your gametes across the top and down the side. Then you fill in your possible genotypes of your offspring, which are in the boxes. So what you do is you just pull in a sperm cell and an egg cell, and you have fertilization to get back up to your 2 letters. So we take a big T and a big T, pull them into a box, we get an individual offspring of big T, big T. We take a big T, little t, pull them into the box, little t, big T, pull them into the box, little t, pull them into the box. So you have your 4 possible offspring inside of your Punnett square. Okay, so that's great. We have a bunch of letters in a square. So again, we need to figure out what this actually means, because science isn't just doing the experiments and collecting data. We need to come up with conclusions. So in step 5, you're going to determine the proportion of genotypes or phenotypes, and this is kind of depends on what the question is asking you, so we're going to be doing both of these. So sometimes you have to figure out the ratio of genotypes. So we have the genotypes, which again are the genes, so the letters inside the box. So we have an offspring that can be big T, 2 of them that are big T, and then 1 that's little t. So we end up with 1 big T, 2 big T, and 1 little t for our genotypic ratio. So that tells us what types of genes our offspring have. But probably the more important questions, they want to know what the offspring will actually look like, so what their phenotype is. If you remember from previous information, our big T is the dominant trait. So we're going to have big T codes for tall, big T also codes for tall. So we have 3 tall individuals, and then we have the little t that'll end up being dwarf. So we'll have 3 tall to 1 dwarf. So you can see that here we have Mendel's ratio show up again for this monohybrid cross up here. So this is how you use your Punnett square. So refer back to these steps. The steps in the homework are exactly the same. You'll figure out the parents, figure out the gametes, put the gametes on the square, fill in the possible offspring, and then answer the question that is being asked off of your Punnett square. So we talked a lot about this monohybrid cross just looking at one trait. So those crosses are pretty simple. Hopefully you don't have too many problems with them. The next type of cross that Mendel did was called a dihybrid or 2-factor cross. So now he's going to look at the inheritance of 2 different traits at the same time. So this is when it gets more fun and more interesting. So he had 2 hypotheses. Either the 2 different genes were linked, so that variants found in the parents were always inherited as a unit. So basically, the offspring will look almost exactly like one of the 2 parents, and not as a combination of any of the parents. The other possible hypothesis that he came up with was that the 2 genes are independent from each other and they'll be randomly distributed to the offspring. So you can kind of guess probably which one he supported with his dihybrid crosses. But just to show you the difference, so on the left-hand side we have his first hypothesis where the 2 genes are linked so that the offspring will match one of the parents. So if our parents are yellow, round, or green and wrinkled, our F2 generation will be yellow and round or green and wrinkled. So that just means that the 2 traits are inherited together. The big Y, big R stay together, the little y, little r stay together. On the right-hand side, we have the 2 genes are independent and randomly distributed. So in our F2 generation, we get all possible different types of combinations. And again, you probably can guess which one he supported with his crosses. It was the one where the 2 genes are independently assorted from each other. So when he collected his data, he found that he could get all possible different combinations. He could get yellow seeds that were round, yellow seeds that were wrinkled, green round, and green that were wrinkled. So you get all those different combinations. So from this cross, he came up with his second law, his one really important idea. This is that these alleles in your 2 different genes are going to sort independently of each other. And the F2 hybrids, they show a phenotypic ratio of 9:3:3:1. And you'll find this out when you do a few problems. You'll learn more about that ratio. But right now let's just focus on Mendel's law of independent assortment. So this is where the alleles of the different genes sort independently of each other during this gamete formation. So what you want to do is you want to use something called the FOIL method. If you've taken a math class, you've probably used this. It may or may not have been called the FOIL method, but it's a very similar and common method in math and science that's used quite a bit. So what happens is that we have a genotype of one individual shown up here. So we have big R, big Y, little y. So just to kind of explain this, the R's represent the seed texture. And remember, For seed texture, you need 2 letters. So we have a big R and a little r. The Y's represent seed color, and this is a different trait, so again we need 2 letters for that trait. So we end up with a total of 4 letters up here, and that is an individual's genotype. So when you create the gametes, you want to create all different possible combinations. One thing you need to remember is that you need 1 R and 1 Y. You don't want a gamete with 2 R's, you don't want a gamete with 2 Y's, because then you'll have gametes that are missing one of your 2 traits. So you need 1 R and 1 Y in every gamete. I don't know how to stress this anymore. Students still manage to mess this up, and I'm not sure why. But the FOIL method seems to help out with this a little bit. So the FOIL method, what you're going to do is you're going to take the first R and the first Y and put them together in a gamete. So you'll have a gamete that is big R, big Y. The O stands for outside, so you'll take your outside letter, which is a big R, you'll put it with the other outside letter, which is the little y. So you'll have another gamete that's big R, little y. The I stands for inside. You'll take your 2 inside letters. So we'll put a little r with a big Y. So you have a gamete that's little r, big Y. And then the L stands for last. You'll take your last r and last y. So little r and little y, and put them together in a gamete. So there we have all of our different possible combinations. But remember, you need a big R, or you need an R, and you need a Y in every gamete. Okay. Otherwise, again, you'll be missing one of your 2 traits, so you need a allele from each of your 2 traits in your gamete. Okay, so we'll look at these 2 laws in lab quite a bit. And there's also homework problems that you can work on, and you can also use your book to kind of get into more examples of doing different crosses. So we looked at Mendel's law of segregation. So the 2 copies of your genes are going to segregate or separate from each other during the transmission from parent to offspring. So that's where you take your one T and separate it from the other T in the example we went through. Mendel's law of independent assortment, this is why you're going to use the FOIL method if you're looking at 2 or more traits. So alleles from different genes are going to assort independently of each other during the gamete formation. Okay, so this first part, it's probably the most important part of the whole chapter, so it's a little bit longer lecture. So this was Mendel's 2 laws of inheritance. In the next part of the chapter, we'll look at these other interesting parts of inheritance.